Thank you so much to Cure Duchenne for this invitation to speak today. My name is Mark Stahl, and I'm Director of Clinical Development here at Avidity Biosciences. I am excited to share information about us, our science, and our work with the Duchenne community with you today. These are our forward-looking statements. At Avidity, we are committed to improving the lives of families impacted by rare diseases, including Duchenne muscular dystrophy, through the development of new drugs. We are a biotechnology company based in La Jolla, California. We are grateful and honored to serve the Duchenne community and have the opportunity through events like this to hear the perspective of people with Duchenne and their families and their caregivers. We consider patients to be at the heart of everything we do. Our commitment to rigorous and innovative science is matched only by our passion to see improvement in the lives of people affected by rare diseases. We are also grateful to Cure Duchenne and other advocacy groups that help us connect together. Today, I'd like to talk about the drugs we make at Avidity, how they work, and how we plan to use them in treating Duchenne. Our drug technology is called the AOC, the Antibody Oligonucleotide Conjugate. AOCs provide a new way to deliver drugs directly to affected cells, like muscle and heart cells. The AOC platform combines proven technology of existing drug types that are well understood for each of their important properties in drug development. The first part is the antibody. An antibody is a naturally occurring protein made by your body to help fight infections. Antibodies are commonly used in drug development because they can be made to target almost any protein, including those that are different between one cell type and another. Antibodies are well characterized for their safety and are the foundation of over 100 approved drugs, many of which are used in chronic conditions. At Avidity, we use the antibody as a vehicle to deliver our drugs to muscle cells. The second part of our AOC platform is an oligo, or oligonucleotide. An oligo is a short strand of DNA or RNA. Oligos can be powerful drugs because they can change gene expression, or how the body makes a protein by targeting RNA. There are currently multiple approved oligo-based drugs, many of which are used in disease-modifying treatments. At Avidity, we use oligos in our AOCs that are created specifically to target the genetic cause of disease. For muscle diseases, the antibody component of the AOC enables targeted delivery to muscle and heart cells, and in turn, improves the power of oligos. The antibody has the unique ability to recognize and stick to a specific protein on the surface of these cells that delivers more oligos that change the gene expression. Specifically, the antibody binds to a transporter protein called the transferrin receptor 1 on the muscle cell. It is then carried inside the cell where the oligo is released. You can think of our antibody as a bus that's taking its passengers to a very specific bus stop and then letting them off to go to work. In our case, that bus stop is a muscle or a heart cell, and the passenger's job is to help make dystrophin. The combination of antibodies and oligonucleotides that make these drugs, called AOCs, result in the ability to target specific cells in the body, the heart cells and the muscle cells, and modify their gene expression, as we are working on with dystrophin. This is allowing us to develop new treatments for diseases where current therapies are limited, all in the service of improving the lives of people affected by these conditions. Currently, Avidity is developing 3 different drugs that work via the exon skipping technique that has been discussed earlier in the meeting, and I will explain a bit later. These are drugs designed to treat the root cause of DMD in individuals who are amenable to skipping of exons 44, 45, or 51. Our first program is called AOC 1044, and it is designed to treat individuals whose gene mutation is amenable to exon 44 skipping treatments. We plan to apply learnings from the research and development of AOC 1044 to our other programs in Duchenne. Taken together, we aim to be able to treat about 30% of the total Duchenne population with exon skipping technologies. Here, I will talk a little bit about how our drugs work in more detail. As I mentioned, our oligos work via something called exon skipping. Skipping an exon ultimately results in the production of a slightly shorter but still functional version of that dystrophin protein. Here's how it works. Many mutations that cause Duchenne result in the gene sequence becoming what's called out of frame. This means that when the genetic code is read by the cell, a stop signal is encountered, resulting in the production of little or no dystrophin protein. Exon skipping causes the cell's genetic material to skip that stop sign, restoring the reading frame. This results in the production of dystrophin where before there had been none. Currently, there are 4 approved exon skipping treatments for subsets of the DMD community. Each is designed to skip a specific exon. Particular mutations are amenable to skipping of only a specific exon, if any, so it is important to know which mutation an individual carries. However, there have been some limitations to the currently available therapies. The current treatments are based on the use of unconjugated or naked oligos. They are not attached to any antibody or other delivery vehicle. Without targeted delivery the antibody provides, oligos have inefficient uptake by muscle and heart cells and require frequent dosing. Our AOCs combine antibodies that target muscle and heart cells with exon-skipping oligos. In this diagram, you can see the AOC is on the left. The antibody part of the AOC has a unique ability to find a specific protein, the transferrin receptor, on those muscle and heart cells and deliver that oligo into those specific cells. Once delivered, the oligo enters the cell to modify the gene expression by targeting RNA. By combining the oligo and the antibody, we are able to more efficiently deliver the oligo directly to muscle and heart cells. More oligo is therefore able to enter those cells and increase exon skipping, which in turn can lead to increased production of the dystrophin gene product. Here, I will show just a little bit of the data our scientists have been generating with our technology. In this experiment, we used muscle cells from boys with DMD, amenable to exon 44 skipping, and we tested our oligo. We achieved substantial exon skipping, up to 60% of the RNA produced. In lab studies, AOC 1044 was seen to be very specific for exon 44 with no off-target effects. AOC 1044 also showed a long duration of action, meaning it could stay active in the body long enough to allow for monthly dosing. In this experiment, we used a mouse model in which we could compare the exon skipping ability of unconjugated or naked oligo directly to one which was part of an AOC. This helps us understand the effect of linking the antibody to the oligo on the effectiveness of the drug. You can see that treatment with the AOC led to a greater than 50-fold increase in the degree of exon skipping compared to an unconjugated oligo in a mouse model of DMD. At Avidity, we are committed to partnering with the DMD community to bring forth meaningful treatment options as we strive to continue to partner with the DMD community and incorporate the patient voice into everything we do. We are continuously working with and supporting advocacy groups to ensure that we are continually seeking input from the community throughout the drug development process. By collaborating with key groups like this one and community leaders, it will ensure that we are addressing any gaps in support services and resources. Here are some key takeaways from my talk today. We are very excited by the potential of Avidity's AOCs to treat the root cause of Duchenne. Preclinical and animal studies are ongoing, and in preclinical studies, we have seen increased exon skipping that allows for the production of dystrophin protein with our AOC treatment as compared to naked oligonucleotide treatment. Our AOCs are expected to be administered with infrequent IV dosing, and we are planning to initiate our first clinical trial with AOC 1044, currently in development for the treatment of boys who are amenable to exon 44 skipping by the end of this year. Finally, one of my favorite slides. These are some of the Avidity team members who are the faces of the company and are working hard every day to address the unmet needs of patients and the DMD community. We are an extremely passionate group and love the work that we do. It's our privilege to serve this amazing patient, caregiver, and advocate community. We are always hoping to listen, learn, and connect with the community, so please feel free to reach out to us. You can connect with us at the address at the bottom of your screen, patients@aviditybio.com. Again, thank you for your attention.